Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Yves Gaudin. He's a researcher over in France. There are many French words and that describe his degrees and his university, and instead of me butchering it, I'd rather have him pronounce it. So, Yves, thank you for coming.
Thank you.
Yeah, would you just give a brief background on where you're working right now, and let's talk about your research.
Yeah, in fact, I'm working for the CNRS, which is the National Center for Scientific Research in France. We are a few thousand, 30,000, I think, working in this kind of institution. So I'm a virologist, and I'm working on the viral family, which is the Rhabdovirus family. The two best studied viruses of this family are rabies virus, for, of course, obvious reasons, because it's still killing a lot of people, and VSV, which is vesicular stomatitis virus, which has always been an excellent laboratory model. We are dealing with the entry mechanism, how the virus is recognizing its receptor, how it penetrates the cell by a membrane fusion event, and also how in the case of rabies virus, how it installs its viral factory. And we have characterized the nature, the physico-chemical nature of the factory by showing it's formed by liquid-liquid phase separation. And in fact, we are also dealing with the interaction between this viral factory and the innate immune system, all what the pathway to the synthesis of interferon, and also then how the interferon stimulated gene, the product of those genes, interact with those factories and with the viral cycle. This is more or less what we are doing. Okay. In our team, we are about 10 persons.
Okay, well, let's start with the rhabdoviruses. You said rabies is one of them?
Yeah.
So, okay, are the rhabdoviruses giant viruses, or what's unique and interesting about them at first glance?
Well, in fact, they have always been a good model for RNA viruses. They belong to a large order of viruses, which are made by, of which the genome is an RNA molecule, a single strand RNA molecule, and they are called, they are the mononegavirales because this RNA molecule is of negative polarity. In fact, it is the opposite sense of the RNA messenger, so that when the virus is releasing the genome in the cytoplasm of the cell, it has to be transcribed into RNA messenger by a polymerase, which is brought by the virus. This is an activity which does not exist in the cell, and so that's a huge order of viruses in which there are the measles virus, mumps virus, Ebola is also belonging to this order, and there are several families, and the rhabdovirus are one of this family.
When you say it brings along this ability, is it just part of the base pairs of the RNA strand, or is there a separate is there a separate chemical or molecule that does this transcription?
Yeah. In the viral particle, there is the genome, and inside the viral particle, there is also a polymerase, which is an RNA polymerase, RNA-dependent RNA polymerase, which makes RNA from an RNA template. And this activity, of course, does not exist in the cell. And when the genome is released into the cell, immediately this polymerase, which is brought by the virus, is acting on the genome to make messenger RNA, which have the same properties as the cellular messenger RNAs, so that, the translation machinery of the cell, which is synthesizing the protein, is hijacked by the RNA messengers, which have been synthesized by this RNA polymerase. And in fact, this is common to rabies virus, of course, all the rhabdovirus, but also Ebola virus, which is from another family, the filovirus. There are also the paramyxoviruses, such as measles, mumps, also Nipah virus, which is a very, very dangerous virus for humans. So there are several viruses which are causing deadly disease, which are also in disorder and share the same mode of replication as rabies virus. But also rabies virus, of course, is still killing about 60,000 people each year around the world. Of course, it's much less than the SARS-CoV-2, but it's still significant because it's every year.
Well, okay, so the polymerase that gets released once the rhabdovirus enters the cell, it acts on the host material, or does it act on the viral particle material, the RNA?
So it acts first on the viral RNA, the genomic RNA, the genome RNA, which is an RNA molecule, and it is immediately transcribed into RNAs that are complementary to the genome. And these new RNA molecules, which are transcribed from the genome using the viral polymerase, are the viral messenger RNAs, which then hijack translation machinery of the cell.
Oh, so without the polymerase, the RNA from the rhabdovirus would just be inert inside the cell?
Exactly. You need the polymerase to be inside the incoming virion. The incoming virus needs this polymerase. If not, there is no infectious cycle. And of course, before, and that's the thing that we are working on, for the virus, there is a necessity to release this genome, and this is made by the glycoprotein, which constitute finally the spikes of rabies virus, but of other rhabdoviruses, just as the spike of SARS-CoV-2, in fact. And it is as the spike of SARS-CoV-2 able to recognize a receptor first, and then to fuse the viral membrane with a cellular membrane. And then this fusion event allows, there is then a continuity between the inner of the virion and the cytoplasm after the fusion event, and the genome, together with the polymerase, are released in the cytoplasm of the cell for the subsequent step of infection.
Okay, gotcha. So is the whole capsid endocytosed once the membrane fusion happens, or just the RNA and the polymerase is injected?
If we summarize, first, the glycoprotein, which constitutes the spike all around the virion, recognize a receptor. There are several receptors at the surface of neuronal cells for rabies virus. Then virion attached the cell, and then this triggers an endocytosis event, and the virus enters the cell through the endocytic pathway, but there are still the viral membrane and the membrane of the endocytic vesicle around the virus. So to release the genome, you need this fusion event between the viral membrane and the membrane of the endosome. And then, of course, you induce a continuity between the cytoplasm and the inner of the virus, and then the genome is released into the cytoplasm. And this fusion is triggered for rabies virus and for other rhabdoviruses by the acidification of the endocytic pathway. In fact, in a normal cell, there are proton pumps which acidifies the inner of the endosome. And this acidification event is hijacked also by the virus to trigger a conformational change of the spike of the glycoprotein, which was previously involved in the receptor recognition. But then this spike, upon acidification of its environment, is able to trigger the fusion of the membrane, of the viral membrane, with the endosomal one. And this is very classical. Same phenomenon for influenza virus. Same phenomenon for several viruses, which hijacks the endocytic pathway to enter the host cell. It's a bit different for SARS-CoV-2, but there are several similarities anyway.
So let's talk about rabies a little bit. How does rabies work? The virus enters the cells, and does it lyse them, or does it what action does it take in the human body to cause sickness?
Well, the first event when you get rabies is that in general is that you are bitten by an animal, which is, for example, a rabid dog. Then the virus finds its way. Maybe there are some suspicions that it is able to have some cycles in muscle cells, but in general, it finds a nerve terminus, and then the virus is able to enter into the neuron. And then it will be transported all along the axon of the neuron, and by successive infection of neurons, it will reach the nervous system. And then once the rabies virus is in the central nervous system, then this is finished. The issue of the disease is always the death. This is a terrible disease, in fact. By chance, you can vaccinate after the bite of the rabid dog, because the virus is coming slowly from site of the bite to the brain. But once the virus has reached the brain, this is finished. There is no other issue than the death. But if you've been vaccinated early, you may block this progression of the virus toward the brain. And it is not clear why people are dying from rabies virus. Of course, the several neuronal cells are not functioning correctly. They have some defects, but there is no massive death of neurons. This is probably linked to bad functioning of some neurons which are unable to behave normally. What we see very often is extreme agitation of the patient. And you see also people are extremely aggressive. So you may have some symptoms of very, very strong aggressivity of the person who has been contaminated by rabies virus once the virus has reached the brain. But also sometimes you observe paralysis. People cannot move. And of course, this is always the end of rabies virus. At one point, there is a paralysis which installs. And then you just cannot breathe, and then you die, of course.
That's terrible. Have there been any advances in rabies treatment over the years? Well, is there really any treatment for it or no?
Well, there is no other treatment for the moment than the vaccine. And of course, you are not going to vaccinate everybody, to give the vaccine to everybody, because in fact, in most of the countries, there is no more rabies. The rabies disease has disappeared. For example, in Europe, there is no more rabies disease, except for people who may have spent holidays, for example, in Africa or in Asia, and have played with animals. This is something people often forgot. When you are in some countries, you don't play with animals. You don't play with dogs. You don't play with wild cats. You don't play with, of course, with wild animals. You just let them away. This is a good behavior to leave those animals away, because there may be some virus, and then they can transmit it to humans. This is also the case probably with some coronaviruses, but in any case, rabies virus is. So in fact, when you've been bitten, the only way is if the animal is proved to have rabies disease, then the only way is to be vaccinated. That's very fast, immediately. And you have also serotherapy. There is an injection of antibodies which are directed against the virus, just to try to block the progression of the disease. And if you can block the progression of the disease, the virus is not entering your central nervous system, then it's okay, you will survive. But if despite treatment, it reaches the virus is reaching the brain, then there is no other issue than death. So this is a terrible, terrible disease. But there is no molecule, because it's a bit like, for any virus, and we are experiencing this for coronavirus, we need specific molecules. And this is a lot of money which needs to be invested. And in fact, with rabies virus, to speak frankly, and this is a fact. This is not maybe this is not an ethical point of view, but there is no market. There is no market, and rabies virus is always in poor countries in general. There is no money, so certainly for the pharmaceutical industry, there is no market for rabies virus. Now there is a market for coronavirus. Probably someone who is getting an efficient drug against coronavirus, this is interesting. But for rabies virus, there is no market.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So why is there no treatment? How come not enough appears to have been learned in order to treat rabies after all this time?
So you mean why there is no treatment against viruses in general? Or? Yeah.
Well, I mean, viruses in general, but especially rabies. Why? I mean, can't we identify the polymerase, maybe block it?
Yeah.
There's got to be some treatments.
Yeah, I understand. So, well, of course, the polymerase is known. The structure of the polymerase is known, as well as the structure of coronavirus and a lot of viruses. But first, well, it's easy to find some molecule which blocks the virus, in cell culture or when we are making experiments. But in general, those molecules do not work well when you go into an animal or into a human being. Of course, knowing the structure of the protein gives a rational basis to imagine molecules that can block, because all these, for example, the polymerase, you can think about a molecular automate, and you can try to block this molecular automate. But in practice, it's not that easy. And you've heard certainly about the Rhabdoviruses, for example, which was a molecule which was thought to have a pan-viral activity against RNA viruses. But it seems that this molecule is not really efficient, neither for the coronavirus, nor for Ebola, nor for probably rabies virus. Of course, this may be tried, but in fact, if we think about the AIDS, all the molecules that we have against HIV have been specifically designed against HIV. And you have to spend time for that. And once again, and it's a lot of investment for a few you make, for some molecules that you identify in a screen in cell culture. Only a few of them will go to an interesting compound. And then it's, again, a lot of tests. And for several hundreds molecules that are analyzed, just one or a few will get on the market. So I understand that it's very complicated for HIV. Of course, there was a market, and in rich countries furthermore. So, but it's very difficult. It's a lot of work. It's for rabies virus. It will take certainly some time. Maybe things will change with this pandemic of coronavirus. Maybe people will understand that it's interesting to have specific molecules against viral families, so maybe against rhabdoviruses, against filoviruses like Ebola, against coronaviruses, against flaviviruses like dengue virus or Zika, but mentality have to change first. So it's a lot of work. It's not that easy. It's a lot of investment with a lot of disappointment with some molecules.
Well, so in addition to, we spoke about rhabdoviruses a bit. You mentioned they have this RNA polymerase, but what about Ebola, the filoviruses, and some of the other ones? Do they have any interesting or unique mechanisms that help coordinate their activity inside cells?
Well, in fact, all the viruses from the mononegavirales order, so Ebola, they have a very, very similar polymerase. So, of course, it's not exactly the same, and the active site may be slightly different, but they have the same activity. And the polymerase is able to synthesize, of course, an RNA molecule using an RNA template as a model. They can also add some specificities to the RNA they are synthesizing, which makes the RNA they synthesize, which makes those RNA looking like messenger RNAs. So once again, allowing the virus to hijack the ribosome, the translation machinery of the cell. They have all these same kind of activities. So this is there are some similarities, and also their mode of replication is very similar in terms of for rabies virus, Ebola, mumps virus, measles virus. come through the same stages to replicate their genomes. So there is a lot of similarity. What we learn from one is very useful for the other, particularly when we have demonstrated that for rabies virus was able to form viral factories having liquid properties, and we can come back to what we mean by this word, liquid. So we demonstrated that for rabies virus, but it was also then demonstrated for measles virus. And also there are several data which indicates that it's the same for Ebola, for respiratory syncytial virus, the virus which is causing bronchiolitis. There are a lot of similarities. They interact with the cell in a very, very similar mode.
What does it mean they have liquid-like properties?
So, liquid properties. So, what's interesting with that is that the viral factory, when I say viral factory, this is the place in the cell where the virus is synthesizing the RNA, the messenger RNA, but also the genomic RNAs, because at one stage, the virus is going to copy its genome to make new genomes. And it's the same polymerase which we've talked about, which is doing the job to synthesize new genomes and to amplify the infection by making new genomes which assemble and make new variants. But then, in fact, this occurs in a compartment. And this compartment is just like oil in water, This compartment in the cytoplasm, it's just like oil in water. When you put oil in water, you form a spherical droplet of oil, and oil is unable to mix with the water. There is what we call a liquid-liquid phase separation. We separate to liquid. And it's exactly the same. The liquid factories of those viruses is unable to mix with the cytoplasm. So somehow the virus inside the cell is keeping its identity. There is this debate about viruses, about are they living organisms? And there are several facts which indicate that viruses are not living organisms. They do not have metabolism. They are unable to synthesize their own proteins. They use machinery of the cell, etc. But there was also this idea that the virus is perfectly identified outside the cell. This is the viral particle that you see in electron microscopy, that people have seen a lot of coronaviruses as viral particle outside the cell. But once the virus is inside the cell, once the genome is released inside the cell, there is this idea that the virus is losing its identity. Its genome is mixed in an ocean.
It's like a virus cell. It becomes
It's mixed with the content of the cell. But in fact, this is not the case. The virus is able to keep its identity. It is making viral factory. And this viral factory, just as oil is not mixing with water, this viral factory is not mixing with the rest of the cell. This is the viral compartment. This is the place where the viral identity is inside the cell. Of course, it can exchange some material, but this is based on the properties of this material, and he has selected, and is able to select the molecule he needs just to build new variants, in fact. But of course, the cell is also fighting against infection. And you may imagine that cells have developed strategies to disrupt this factory. And I think this is a next step in virology to identify all those proteins, all those cellular proteins which may act against this viral factory.
Has there ever been a case observed where a cell is infected with a virus and the virus leaves and the cell goes back to normal without infection?
Oh, yes. You mean, of course, there is a possibility for the cell to eliminate the infection at early stage, even for any cells, because, there is all this interferon signaling, and some cells, when you have an infection, immediately when the cells are infected, they produce interferon. And in fact, this interferon is circulating in your body and is preventing also the other cells to be infected, because once they have been in contact with interferon. There are some pathways inside the cell which indicate to the cell that they need to synthesize some molecules which have antiviral activity. And of course, if a cell has already all the weapons ready against the virus, when the virus is arriving, the virus will be disrupted. And or the genome will be disrupted, or there will be some blockage at some stage of the infection, and so that the virus will not be able to have a productive infection. And the cell may survive, of course, in this case, because the cell is ready to fight.
When a virus takes up shop inside of a cell and it creates what you call viral factories, how much of the cell's energy and machinery is dedicated to making more virions versus running metabolic processes?
Yeah. So if you take, this may depend on the cell, and of course, this may depend on the virus and on the nature of the viral factory. For example, for a virus we've already talked about, which is vesicular stomatitis virus, which is the causing of rabies virus, it is known that when the virus is entering the cell, it is able to block all the synthesis, all the transcription of the cell. The cell is unable to transcribe its own messenger RNAs. So at one point, there will be only viral messenger RNAs. There is really a hijack of the host machinery. Just there is the cellular transcription is blocked, is stopped. There is also some similar data, for example, for the coronavirus. For the coronavirus is blocking the translation of the cellular messenger RNAs. So they are synthesized, but they are no more translated into proteins. So this will depend. Of course, the virus needs also some cellular proteins. So there are some kind of balance between what you absolutely need to block and what you want, what the virus needs to block, and what it is keeping, it is keeping functional. So there is a balance, and this may depend from the virus to another. But in general, the virus is trying to block all the innate immune response because, as I told you, this is a way for the cell to fight the infection. And it is also trying to hijack all the translation machinery because it needs the translation machinery to make new viral particles. And then, of course, it is also known that all the cellular energy, this exchange money, which is the ATP, is also produced by the cell for the virus. The virus is really it's a very strong parasite. It is using all what the cells is offering to him.
Interesting. What do you think could be intervened in terms of the viral factory at that stage? What could be done that would cause the viral factory to slow down or stop or throw a wrench into the works?
Yeah, I understand. So clearly, let's say 3 or 4 years ago, no one was aware that these factories had liquid properties. So this is really a change of paradigm, and we may imagine first that we may find some small molecules which destabilize. If you put oil in water and then you put detergent, and then the oily droplet is mixing with the water thanks to the detergent. Of course, it's not the same kind of interaction. In fact, in oil in water, it's hydrophobic interaction, which is keeping the oily droplet is disrupted by the detergent, and the base of the interaction between oil molecules is the hydrophobic interaction. In the case of the viral factory, it's another kind of interaction. So detergent will not be useful for that. But we may find some molecules which have properties to disrupt weak interactions between the viral proteins, which are keeping the homogeneity of the viral factory. We may find some molecules. And in fact, also, that all those RNA viruses are inducing fever. And this phase transition, these liquid-liquid phase separation are highly sensitive to temperature. And it's possible that increase of temperature when we have fever may destabilize, may slightly destabilize all these factories. And fever might have been a kind of evolution of our organism to destabilize all those liquid structures that are formed during viral infection. That's one possibility. But of course, this opens new pathways for research and also the subtle interaction between these viral factories and all the proteins that are involved in innate immunity also opens some new areas of research which may lead to new treatment against those viruses.
So why is it important to look at the dynamic of hydrophobic and hydrophilic interactions? What will that tell you?
To keep the structure of these spherical factories, you need weak interactions. You need weak interactions between viral molecules. So we have identified some of the proteins which are involved in those weak interactions, but the molecular basis of this interaction is still unknown. So we need to go to, and it's difficult to investigate that inside the cell. So what a lot of people are trying, not only with this kind of liquid viral factories, but all novel organelles in the cell which have liquid properties, people are trying to identify those forces, those weak interactions between the components. And for that, in fact, they want to reconstitute the system into a test tube. And then one of the best tool to investigate those interactions is NMR, nuclear magnetic resonance. So this is the traditional approach. So you can have an idea of, because NMR can identify these weak interactions. That's a possibility. But also there are a lot of other approaches which are currently developed to go further in the understanding of the molecular basis of these weak forces between proteins, but not only proteins, but also RNA molecules, because all those structures also contain RNA molecules. And yes, this is something very intriguing. And also there are several questions around this subject, because at one stage, the virions, which have just been synthesized, they have to leave this viral factory. How do they leave this factory? How do they go away? Because somehow they are happy in this structure. They have good affinities for this structure. So there are a lot of questions. And also the understanding of the weak forces may help to understand why some cellular proteins can be sequestered inside the factory. And they are there because the virus is needing those proteins, and some others are completely excluded. They cannot enter the factory. And so here again, there are some principles, some molecular principles that we need to understand if we want to go further in this domain.
Very good. Yves, what's the best way for people to find out more about your work? Where can they go?
I think the article that we've published in Nature Communications in 2017, which is accessible to everybody. So this is good about those viral factories. There are a lot of movies showing the dynamics of those structures inside the cell. And I think it's very spectacular to see that once a virus is entering the cell, it makes a lot of things. And in fact, we have built recombinant viruses with fluorescent proteins so that we can follow all the movement of the factories. And you will see that the factories are very, very round, very spherical. And this is due to the fact that they are formed by liquid-liquid phase separation. And you will see also some of those factories coming into close contact and fusing together to make a bigger one. And you will see also that upon encountering an obstacle, they reversibly develop, showing that indeed they are made of, they are liquid and not solid or gel-like structures. So the article published in Nature Communications and the movie are available because Nature Communication is an open journal, and everybody can see the movies, which are, I think, very spectacular.
Oh, nice.
Yeah.
So last thing, I just want to repeat this. So you said that instead of gel-like structures, they're actually liquid-like structures?
Yeah.
How can you tell? I mean, is it just by microscopy you can tell the nature of the structures, or how could you tell?
There are several experiments which are key. First, as I told you, when two viral factories, when into close contact, they fuse together and relax toward a spherical structure. And if you have two droplets of oil into water, they will do the same. So this is an argument to say that this is a liquid structure. And of course, what we know that there is no membrane around. So it's because if there are some membranes around, you may imagine that the membranes are fusing together, but there is no membrane. We have looked in electron microscopy. There is no membrane around those structures. What we see also is that, as I told you, they can reversibly deform. So when there is an obstacle, and the obstacle inside the cell is, for example, a molecular motor, which is pulling a vesicle, and in one of the movies, you will see a spherical object, which is a vesicle, which is probably pulled by a molecular motor, which is crossing the viral factory, and the viral factory is deformed and reformed after the passage of the vesicle. And what we can also see is that those structures are extremely sensitive to the physico-chemical environment. And there is also a movie where we have performed a hypotonic shock. So we put the cells into a medium with a lower salt concentration. And then what you see, of course, the cells have a tendency to swell, but immediately the structures disappear. That's just immediate, which is of course not possible with a gel or with a solid. This is just a matter of seconds. And then when the cell is able to recover its normal homeostasis, then you see the viral factories which reappear inside the cytoplasm. And finally, we've used an experimental approach which is called fluorescence recovery after photobleaching, FRAP. The abbreviation is FRAP. And in fact, we use, once again, those fluorescent viruses with fluorescent proteins, and then we burn a small volume inside the viral factory, and we see that when we burn the proteins, it is no more fluorescent. And of course, this protein is progressively exchanging with the other molecule, and you see that quite fast you recover the initial fluorescence level. In fact, we burn the whole factory, and then what we see is that there is an exchange with some proteins which remain outside the factories, and they go very fast inside. If it was solid, then probably the exchange would have been much slower, and sometimes if it was a solid, there even might have been no recovery of fluorescence. So this is a technical approach. But the fusion of vesicles, their ability to deform, their sensitivity to the physico-chemical environment, and their spherical aspects are good arguments in favor of their liquid properties. But when you see the movies, you have very, very little doubt about that.
Very interesting. Okay. Well, I can't go see regular movies right now, so I might as well watch virus movies. That'd be at least something to watch, you know.
Yeah.
Well, very good. Well, Yves, thank you for coming on the podcast. I really appreciate it.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.